All right. Can everyone hear me? All right. My name is Jasper Chao. I'm a PhD student at Penn State University. I'm here to tell you a little bit about bacterial gene function in vulture microbiomes. So right off the bat, let's just get into the key question that I wanted to ask here, which was whether vulture gut microbiomes aid their host's unique diet. Vultures, of course, are well known for their unique diet, consuming carrion and rotting meat. These are hosts to pathogenic bacteria and toxic compounds. So the question is, how do vultures deal with it? Let's move on. Previous studies looking at vulture gut microbiomes, specifically this one by Rogenbach et al. In 2014, found high concentrations of Clostridia and Fusobacteria. Fusobacteria are generally associated with, or both these species are anaerobic, or both these taxa are anaerobic. And Fusobacteria are associated with causing colorectal cancer as well as stomach ulcers. Clostridia is a genus that's more well known for containing the species botulinum and tetanus, both causing their own respective diseases. And this is great. It's super interesting. However, the study did have its own limitations. It only told us about diversity and community composition, which is interesting, but it doesn't necessarily tell us anything about the bacteria's function in their ecosystem. I would say it's mostly speculative, and it's quite limited in scope, being single gene 16S amplification, which have its own problems, mainly with running the risk of PCR bias. So in our study, we relied on shotgun metagenomics, no amplification. We get access to the genes, so we get to avoid both of these issues. In regards to why we're studying vultures, generally, they're very efficient scavengers. There are other scavengers that exist in the world, wild dogs, rats, crows, and ravens. But vultures kind of run the pros and cons of both, they run the pros of both all these other species without really running the risks. They can fly, so they can cover vast distances and kind of find carrion all over the place. But they also interact with humans much less, so there's much less of a risk of disease and transferring things to humans and throughout the environment. So the question is how they're able to tolerate these bacteria, harmful bacteria, toxic compounds, and how do they remove them from the environment. We know that they're exposed to a plethora of bacteria through their face as they enter whatever carcass that they're consuming. But by the time all that food is passed from their face through the digestive tract, the diversity of species of taxa has decreased significantly, and so has the risk of disease. Mostly you just see risk of, I believe this is, like stomach ulcers and like inflammation. Yeah, so sample collection occurred in kind of the northern part of Florida and the southern part of Texas, kind of by Gainesville and Alice in their respective states. This was done by a collaborator at the Smithsonian Institute, Gary Graves. And we collected samples from each of the two species, black and turkey vultures, along 4 locations along his intestinal tract, collected along the stomach, the duodenum, jejunum, and colon. Duodenum, jejunum ended up being mostly host DNA, 99%, so we ignored it for the purposes of the study. Black vultures were live trapped and euthanized with CO2 gas, and Cathartes aura, turkey vultures were shot and bagged at roosts, and samples were stored in RNA later and frozen until they were ready for sequencing. Just getting to relative abundance quickly, I just wanted to show that we found similar results to the previous study in 2014. We see high concentrations of Fusobacteria and Clostridia. Clostridia are included in the phyla here, Bacillota A. There are some differences between the species that you can see, but primarily the differences as you can see are between sample types. So turkey vultures in their colon contents generally contain more Pseudomonada. This is a group that contains Acidithiobacillia. These are bacteria that are known to extract metals such as iron and lead. black vultures and their colon contents tend to contain more Campylobacter, and this group contains Helicobacter and Campylobacter. Both these are known for causing stomach ulcers in humans. Other general patterns that you can see are more are Clostridia, which are obligate anaerobes that contain tetanus and botulinum in the Bacillata A samples. And getting into the diversity that we see in these samples. So just a breakdown of like each of these different diversity metrics. Richness is just a measure of how many total species you see in the samples. Neutral diversity does not consider genetic variance, so they don't have any effect on fitness. Phylogenetic diversity considers the branch length of the phylogeny, so just considers the relationship of the taxa that you're investigating. And functional diversity concerns things that the organism does in its ecosystem. So overall, turkey vulture colon contents are more diverse than the black vulture colon contents, but you do see the reverse when it comes to stomach contents. And turkey vulture colon contents tend to be more diverse than their own stomach contents. This is the opposite is true in black vultures, except when you consider functional diversity. This could just be because in black vultures, the colon contents need to be generally a little bit more specific. It's a slightly more anaerobic environment, so there's just more bacteria there, might just need to rely on less oxygen. So this is functional diversity, but now we're looking a little bit more at beta diversity, and we're just kind of clustering the sample types now. So you can see that there is not necessarily clustering in samples, but there is distinct grouping. The colon contents and stomach contents tend to differentiate based on their functions, and so do the samples within each of the black and turkey vultures. It's a relatively odd distribution of points in this ordination, but it still shows a general pattern of separation. The next thing we were able to do was actually create a functional ordination of the phyla that we identified. You can actually see some clustering of some bacteria, specifically with Pseudomonada, Fusobacteria, Bacteroidota, and Campylobacter. So these taxa probably provide similar functions within each of their ecosystems. One limitation is that we did not include specific sample types or species, which would have maybe been interesting. Something else we were able to do was show how many genes were present within a specific metabolic pathway. So this grid pattern in the center here, the darker blue values just indicate that more genes involved in a metabolic pathway are present within samples or within the taxa that were identified, and the lighter color, if it's white, means that there's a complete absence of those genes involved in the pathway. And so in general, this just tells us the functional capacity of each of the taxa that were identified. So if we take that knowledge and apply it here, you can see some patterns, but mostly what I want to point to here is this thin line of genes that were present in nitrogen compound degradation. This one here, in amino acid degradation. These genes were involved in serine breakdown, which is an amino acid. I'm hypothesizing at this moment that this amino acid is highly present in toxic compounds, which is why they might be more expressed than other genes in the same kind of metabolic pathway. And you can kind of see a similar thing in this nitrogen compound degradation. And I believe the gene for this pathway is for breaking down GLC-AN, which is important for energy extraction. And sorry, the previous graph was just comparing the two different species that we found. And this one is more, you can see the sample types side by side. And there aren't general patterns between the two different sample types, but you can still see the same expression or same presence of genes. So for main conclusions, I want you to take away from this is that we can see some differences in functions between microbiomes, mostly between the microbes that were identified. But you can also see presence of specific genes of interest, so like those ones I mentioned about amino acid breakdown, the serine and the GLCAM. Future directions, I mean, having these genes of interest are great, but actually like seeing gene expression would be even better. We were able to collect some RNA. We are still waiting to get that back, so working on this transcriptome project would be a pleasure. It would be great. I also mentioned earlier that some of the samples were mostly host DNA. Usually that would be something that we couldn't really work with, but we've been kind of thinking that maybe we could use these samples to tease out some population genetics or population structure in the host species, kind of turn that weakness into a strength. I should have a thank you slide here, but I must have gotten deleted at some point, so I just want to thank my collaborators at the Smithsonian, Gary Graves, members of my lab of Monica Medina's at Penn State University, and our collaborators at University of Copenhagen, Anton Alberti and Tom Gilbert. And I'd also like to thank the NSF for funding this project. Yeah, I've asked for some people's opinions on like what the shape of the plot could potentially mean. I didn't really get an answer, but that is something that they suggested. And it's something I'm going to look into probably after this conference. Yeah.
The host species you looked at were are there specific taxonomic group compared to other cultures?
They're closely related. They're not. Yeah. No. So these are. Yeah, so these are New World vultures. They're genetically distinct from Old World vultures.
Yeah. So my question is, do you get access to samples from, these birds that are taxonomically or evolutionarily distinct from the ones that you examined that have a very similar histories, similar foraging habits, for example, or just look for similarities in their microbiome?
So I would love to do that. I would like to mention, though, that most vulture species, well, not most, maybe half of all vulture species are endangered or threatened. Specifically, a lot of vultures from the Old World they've reached that IUCN status. So kind of collecting highly invasive samples from them might not really be the most feasible thing, especially with the methodology that was used to collect these samples where they were killed and then sampled. It's not really feasible, but maybe something more in like a captive environment would be more possible. And that is something that we've looked into with working with zoos and working with fecal samples potentially, but that might not be necessarily the most accurate representation of gut microbiome. Yeah, birds is hard. I'll be quick. So they have one hole that they kind of excrete everything out, so a lot of things get fumbled together. Yeah. Thanks.
Hello, my name is Markella, and today I will be speaking about my PhD project, which uses museum specimens to study the evolution of the oral microbiome in mammals. So I guess most of us here in the room are aware of the increased focus on microbiomes within the field of ecology and evolution. Some examples, like we know that microbiomes can help their host metabolize nutrients, detoxify their diet, regulate their temperature, and even confer some disease immunity. Clearly, all these processes are important to a species adapting to a new environment. However, the majority of these studies come from the gut microbiome. The oral microbiome, on the other hand, is much less studied. However, we know that it is implicated in Alzheimer's disease, diabetes, cardiovascular disease, and lots of other conditions. So it is clear that it has a systemic role on its host, but these studies also seem to be focusing on clinical applications. There's no obvious way why the oral microbiome would only be causing diseases. It may as well be able to act in an adaptive way, but we just haven't been looking hard enough. And indeed, in a previous study from our group, we found some evidence that suggests that the oral microbiome may help wild animals adapt to their environment. We found that the oral microbiome of mountain gorillas is enriched in bacteria that can reduce nitrate to nitric oxide. And nitric oxide is a compound that is involved in vasodilation and is also found in high levels in the populations, in human populations at high altitudes. So this suggests that the oral microbiome may help these animals adapt to their high altitude environments. But of course, further research would be needed to verify that. But I hope that even so, I have made a bit of a convincing argument that the oral microbiome, although it doesn't have as much hype as the gut microbiome, it's still relevant within ecology and evolution. The issue is that even more than the gut microbiome, the oral microbiome is very hard to sample from wild animals. And this is where this fantastic material comes into the picture, Densocalculus. In summary, Densocalculus, which is this black deposit that you see on the teeth of this animal, is a calcified form of dental plaque that periodically mineralizes while the animal is still alive and traps the DNA from the microbiome inside, preserving it so then it can be sampled from museum specimens, sequenced and analyzed using metagenomics. So my aim for my PhD project is to understand which factors shape the oral microbiome of mammals, and specifically focus on ecological factors like diet and habitat, as well as phylogenetic history, to see which are the ones that are driving this differentiation. For this, my colleague Johnny and I had to tour around Europe and collect Densocalculus samples from different museums. And then we went to this ancient DNA lab to Uppsala University in Sweden to process them and prepare ready for sequencing libraries. So after this, we sequenced almost 400 samples from 29 species, and together with a few already published metagenomes, this brought us up to 35 mammal species in this data set, and that represents a good chunk of phylogenetic as well as dietary diversity for mammals. And this data set includes 3 marine mammals, the orca, South American sea lions, and dugongs. So starting with the first question, I was interested to investigate how the oral microbiome differs between taxonomic orders and diets. For this reason, I focused on a subset of the total sample set that includes 3 deeply sampled taxonomic orders: artiodactyls, carnivorans, and primates. These 25 mammal species also represent a diversity of diets. So we have both herbivorous and frugivorous artiodactyls, herbivorous and frugivorous primates. We also have carnivore representatives that could be classified as omnivores and also fully carnivorous. And we also have some orca samples which they fall under the artiodactyl clade, but they are actually, as we know, a marine carnivore. So some results, basically using an NMDS ordination to visualize the differences in microbiome composition. We find that species loosely tend to cluster by taxonomic order, although it's not very clear clusters, but we see the artiodactyls, the primates, and the carnivorans kind of clustering separately. And if we split these orders according to their diet, we can see that diet as well explains part of the variation. Here I have in dashed lines the herbivores and in solid lines the frugivores. These observations are further confirmed by Permanova test that shows that both taxonomic order and diet have a small but significant effect on oral microbiome composition, about 2.5% of total variance, each of them. I also try to identify microbial taxa that differ in abundance between host orders and diets, and here also the abundances of some of the most interesting microbes that I identified as differentially abundant between these host groups, and have the 3 taxonomic orders as columns and the diets in the rows. So first off, this microbial genus Passerella is, we find that it's enriched in the taxonomic order of the carnivorans. This kind of makes sense. Passerella is a known zoonotic pathogen that is found in domestic cats and dogs. But interestingly, it is not enriched in orcas. So this suggests that it is not so much associated with the carnivore's diet as much it is associated with that specific clade of the carnivorans. Second, we find 3 genera, Codiobacterium, Gemella, and Selenomonas, that are enriched in primates. All of these 3 microbial genera are known members of the human oral microbiome. The first 2 occasionally lead to heart infections, whereas the third one, Selenomonas, has been associated with obesity in humans. And interestingly, Selenomonas is enriched in frugivorous primates. So it would be interesting to see if there's a link between its abundance and maybe sugar consumption that could be causing obesity in humans. Next on, I will talk a bit briefly about the changes that happen in the oral microbiome of terrestrial and marine mammals. For this, again, I'm using a subset of the total data set, 8 species, which include the 3 marine species that I talked about, and their closest terrestrial relatives from this data set. So, as expected, we find that marine and terrestrial species differ in their oral microbiome composition, and Permanova again confirms that both taxonomic order and habitat play a significant role. Again, I try to identify taxa that differ between marine and terrestrial mammals, and we find these 3 genera, Alcanivorax and Brachybacterium, as well as Clostridioides, that are enriched in at least 2 of the marine mammals. The first 2 have been isolated in seawater before, so that kind of makes sense. Clostridioides includes lots of human pathogens, and it's not entirely clear to me why it would be enriched in these marine mammals. And last, I will talk about this ongoing part of my project, which focuses on codiversification. So I will be comparing specific microbial phylogenies to the host phylogenies. And this is a bit that has changed a bit from my abstract because it's just like really recent results. To do this, I first needed to assemble bacteria genomes from the metagenomic reads. And 563 of these metagenome assembled genomes, or MAGs, had a completeness of more than 90% and contamination less than 2%. So those were the genomes I will be focusing on from now on. But for now, I will just talk about this one genus, Neisseria. Neisseria is a common commensal of the oral microbiome and is found in a variety of hosts within our data set, so it was a good first candidate to start with. So here we have on the left the Neisseria genomes, and on the right the Neisseria tree, rather, and on the right we have the host species tree. And using the PACO software, we find that there is indeed significant concordance between the 2 phylogenies. But if we look a little bit closer, there is this kind of deep split of Neisseria into 2 clades. Just for simplicity, right now I'll just talk about that first one that has the most genomes in it. So in that first clade, we find that roughly the Neisseria genomes that come from the same or related mammal hosts are kind of clustering together. However, there is one stark exception, and that is the brown bear. We find that all Neisseria that comes from brown bears clusters separately from the rest of the Neisseria genomes. And that is particularly interesting if we consider that this is the mammal species within this context that has the most distinct diet. It is an omnivore, and everything else there is either some brand of herbivore or frugivore. In summary, considering the oral microbiome composition, we see that there is large variation even within species, but overall taxonomy, diet, and habitat of the host all influence oral microbiome composition. And by focusing on these Neisseria genomes, we can see that there's generally some concordance between bacterial and host genomes, which could suggest some level of co-diversification, but there are discordances that notably reflect changes in diet. So for the next steps, I would like to focus on those Neisseria genomes a little bit more and compare their gene content between different mammalian hosts, see if there is some hint that they could be adapted to that specific environment, and also do this analysis again for more bacterial taxa, because it could be that some bacteria show this kind of concordance, this congruence signal, but others maybe don't. Maybe they're just picked up from the environment and they don't have the signal at all. So yeah, that's it. I would like to thank my lab group, the Kusanski group at the University of Edinburgh. I would like to thank all of you for listening. Sure, yeah.
Great. Like, oral microbiome is something that I thought people should know as well. So we love that. Like, were you able to determine whether the museum samples came from wild populations or from zoo populations? Because I wonder if the differences in diet may have affected what you find in the outcomes.
We did our best. I think I can say that in the vast majority of cases they were wild. I'm not sure if like a captive could have slipped through, but this is something that was prioritized during sampling. From the metadata, you usually can have this information. If we didn't have the metadata to determine that this was a wild specimen, we wouldn't sample it. We had one case where there was a possibility that some macaques had spent some time in captivity, so we didn't use those. Yeah.
Just very quickly, you made the point that some taxa are associated with obesity. I think that's right.
Yeah, in humans, yeah.
Yeah, so, I mean, zoo animals are super fat, generally speaking, so I wondered if there might be a link there.
Oh, that could be something, because they definitely, usually they consume, I guess, diet that is not really the wild diet. So it would be very interesting. We have some domestic samples that we want to compare to our wild relatives. So that would be a next step of the project. But thank you for the question. I don't know if I have time. Do I have time? Sure.
Really cool results. Congratulations. I have one curious question. Do you know what is the oldest samples that were able to get like the DNA?
Not crazy old. So I talk museum specimens, but maybe like 19th, 18th century. John, can you help me with that? I don't know.
The first half of the 19th century.
Yeah, so we don't have like what you would consider paleobiological kind of materials. Yeah. Thank you.
So hello everybody. I'm going to be talking with you all today about the unique microbiome that supports photosymbiosis in the Phrygian heart cockles. So a brief overview of my talk here. First I'm going to tell you all what is photosymbiosis. That's from the last one. Then I'll tell you what is my research question. I'll introduce you to the Phrygian study system that I use. We'll take a brief look at my data. We'll talk about some future directions, and then I will remind you of some key takeaways. So first of all, what is photosymbiosis? So this is a type of symbiosis that occurs between a photosynthetic microorganism, which I will refer to as either an algae or a photosymbiont for the remainder of this talk, and then that lives inside of a heterotrophic host, such as coral or giant clams, all sorts of other marine bivalves, and lots of marine organisms in general. And then the host gives CO2 and shelter to the symbiont, and the photosymbiont gives back its photosynthetic products to the host. This can actually supplement up to 90% of coral's nutrition. This allows all sorts of marine species to flourish in nutrient-deficient environments, and it actually supports a very high biodiversity. So my big question is, how does the bacteriome being the bacterial component of the microbiome, support a stable photosymbiosis? And there is evidence from corals that this is occurring. And you can see in this image on your left here, in the far left, the photosymbionts inside of the gill tissues in the Phrygian system that I am about to tell you guys about. So you might currently be asking yourself, why am I studying this in Phrygian as opposed to coral? Obviously, they're very pretty, but they're also extremely diverse. They are found all across the world, and they provide a really nice closed system, unlike corals. In addition to that, they have a really cool internal and external built-in control that I will tell you about right here. So the tissue types they have four main tissue types. They have the mantle, the gills, the gut, and the foot. The mantle and the gills have a very high algae or photosymbiont load, and you can see those in purple, where the mantle and the gill are pointing at them. And then the gut and the foot have a lower photosymbiont load, with the foot having the lowest photosymbiont load, almost entirely absent. And we'll note that the gut is located behind the foot, just so. The other really cool aspect about this system, so we have those internal controls with low photosymbionts versus high symbionts. We also have this external control because the Fraginae family has diverged into two lineages, where one of them on top here in green is photosymbiotic, and the other one below that in red is non-photosymbiotic. Also, to show you here, we have an outgroup at the bottom, which is Tridagna, which is the giant clams. So interesting evolutionary questions here as well. And my questions really focus on the microbiome and comparing between these two groups, how the microbiome may be supporting the photosymbionts. So to get a better look at this, what I did is I looked at alpha diversity, beta diversity, genus level composition, and differentially abundant strains, with mostly respect to having photosymbionts or algae versus not, as well as some of the animal host species and the different tissue types that I told you about before, the gut, the gill, the mantle, and the foot. So a brief overview of my methodology here. We started with museum specimens that were preserved in ethanol. From Guam are our photosymbiotic samples, and from Panama are our non-photosymbiotic samples. I dissected for those four different tissue types, extracted DNA, sequenced the microbiome for 16S, ran the data through a Data2 pipeline, and used phyloseq for downstream analysis. With that, I ended up with this metadata here. So as I already told you about the sample types for the foot, the gill, the gut, and the mantle, I also, and as you can see, the gill and the mantle are our photosymbiotic tissue types, whereas our foot and our gut are not. And then we also have four different species. The top three here are photosymbiotic, and that's Phragm fragum, Phragm suzensi, and Phragm whitleyi. And then we also have our non-photosymbiotic control species, which is the Trignocardia granifera. So the first thing that I looked at was alpha diversity analysis, and this is the number of different bacteria present in a different group. So what you can see here are a lot of different alpha diversity metrics for the photosymbiotic versus the non-photosymbiotic Fraginae. And you can see that there's not really much of a difference. You can see some of them are higher for the photosymbiotic, some of them are higher for the non-photosymbiotic, and overall not much pattern. This seems to hold for the different species as well as the different tissue types, which is actually somewhat reassuring to see all of these have the same number of different bacteria when the beta diversity analysis does show a difference between groups. So this beta diversity analysis is allowing us to look at the microbiome as a whole for each of my individuals and see how they fall out on this NMDS plot. So this NMDS plot is just like a PCOA analysis, if you've ever done one of those, and it allows you to visualize this in multidimensional space, collapsed to the two most important dimensions, explaining 14% and about 11% of the correlation, respectively. And what's really interesting here that I will draw your attention to is the difference between our photosymbiotic and our non-photosymbiotic. So our non-photosymbiotic is in purple, and our photosymbiotic is in orange and green. We did have to drop off Suzensiae due to low sample, not due to low sample size, due to challenges with the dissections. So anything looking at tissue types I wasn't able to isolate will only be with these three species. And what's really important here is you can see that the gill tissues seem to show the greatest difference. Those are at the bottom is the little triangles there. And you'll notice that the gills, which I told you have the high photosymbiont load, seem to drive this difference. In addition, the mantle, which is located mostly at the top, and then also a little bit down the side. So the mantle at the top is for the photosymbiotic, and the mantle towards the side is the non-photosymbiotic. And you can see that those are also separate, and that is, again, photosymbiont having tissue. And then what's really reassuring is that the foot and the gut seem to cluster mostly towards the top. Some of the gut, you might notice some squares out towards those sides. We suspect that's because they've eaten. But especially the foot having no photosymbiont load and clustering very similarly together supports this idea that it is the photosymbionts that are driving this difference in the microbiome. So given that there is a difference in the microbiome, our next question was, okay, so what's in that difference? So the first thing that you'll notice in the photosymbiotic versus not graph is you'll see that it is dominated by this yellow, and that yellow is these Endoisocomonas. And the Endoisocomonas is prevalent in a lot of marine species. We'll talk about those a little bit more later. The other thing I want to draw your attention to is HIM11, the SUP05 cluster, and the spongy bacterium, as all of these are also found in photosymbiotic organisms in other clades. If you look over at our tissue types compared, you can see in the gill section that this is predominantly Endoisocomonas, which, if you remember from our previous NMDS plot, is most of what is driving the difference between these photosymbiont having and not. And you can see in the middle, that's the Trichinocardia, so that's the one that doesn't have the photosymbionts. So that also has this Corynebacterium, which you can also see at the top of this non-photosymbiotic graph in sort of that beige peach color. And that's absent from our photosymbiont, so that's also something that we are going to look into. And then in the mantle, we also have HIM11 in our fragum again, and that's seen in the blue there. So in both of our photosymbiotic tissue types, some interesting symbiotic species as well. From there, we did some differential analysis. So we wanted to see which individuals are more so in photosymbiotic versus non-photosymbiotic. So I apologize for this chart. I know they're not fun to look at, but I really want to draw your attention to the fact that all of these boxes of red are Endoisocomonas. So the Endoisocomonas are extremely prevalent in these host organisms. And then you can also see that for these Endoisocomonas, there are multiple species, and some of these species are enriched in the photosymbiont having hosts, whereas some are enriched in the not photosymbiont having hosts. And so there's definitely different strains of the Endoisocomonas. While some of them do overlap, they're definitely doing different things for different hosts. After that, we looked at the core microbiome. So this is representing all of my samples being my host organism, that original Fragile sample. And in order to make it onto this list, they need to be prevalent in at least 80% of all of those samples. So again, I will draw your attention to these Endoisocomonas coming up as very prevalent as the top of that core microbiome. We also have the SUP05 cluster and a sulfur-producing or oxidizing bacteria, which is also of interest to us. And then we took a look at the core microbiome for the non-photosymbiotic. This is actually at 90%, so even higher, and you can see it's highly conserved. This might be because it is only one species as compared to 3, but still interesting to see this conservation. Again, these Endoisocomonas make up the majority of the core microbiome, but we also have this SUP05 cluster and this HIM11. So these are bringing up some really interesting evolutionary questions about have they lost their photosymbionts, or are they preparing themselves to take on photosymbionts, or are these just ubiquitous in a lot of marine animals? So we do know a little bit about Endoisocomonas. So we know that they are an established symbiont, and they're found across diverse marine hosts. The coral holobiont is dominated by the family, and their genomes show evidence of carbohydrate cycling and production of proteins and amino acid. They've also likely evolved and diversified with their hosts, and perhaps also with their host tissues, as I've shown in my work. So for next steps, we intend to culture the bacteria that we can from the Fragile microbiome and conduct rescue experiments on the algae, the photosymbionts, by putting them in stressful, like nitrogen poor or reactive oxygen species high conditions, and seeing if adding the bacteriome back will reconstitute the algal growth. And with any success, we will do whole genome sequencing. So some key takeaways here. Fragile with algae have a different microbiome from those without. Tissue types also have unique microbiomes. Differentially abundant strains exist between these two. There's an essential core microbiome, and Endoisocomonas seem to be a key microbiome member, perhaps even lighting the way for the photosymbionts. So with that, I'd like to give some acknowledgements to Ricci, Dan, and Viri for specimen collection. I'd also like to thank Kelly Martin as our collections manager, eBioPackard, and NSF for funding my time. And with that, I have some time for questions. Yes?
What are the algae?
So the algae are the dinoflagellates. They're the Symbiodiniaceae. So they're the same kinds of photosymbionts that are found in coral. There are different clades and different families, but yeah. Oh, we just did it, and right now I think they're called ASV1 and ASV2 from our sequencing, or blue or aqua. We also have a couple folks from my lab here who might know any more, but I don't think we have any official names for them right now. But thanks. Yeah.
Very interesting, thank you. Do you know anything about the effects of ethanol preservation on microbiomes?
Yeah, so I was actually really worried about that as well at the beginning. So somebody in my lab before me has done some work to see if there was any significant differences. And I think there was a little bit of difference, but nothing, most of the microbiome is maintained, just because also it's the DNA that you're working with. Obviously, if you're trying to culture, totally different story. Yeah, good question. Cool. Thank you.
All right. Let's start. The day is almost over. Only two talks left and then some posters. But I'm Senne. I'm a PhD student at the Institute of Tropical Medicine in Antwerp, Belgium. And I will present a part of my PhD dissertation, or what is supposed to become my PhD. And the title of my talk is Parasite Evolution Through the Lens of a Unique Microbial Alliance. Something happened with the outline, but okay. But the thing I want to convey is mainly don't forget about viruses when you're doing research in terms of molecular epidemiology and the evolution of infectious diseases. Why? Because, as most of you will know, viruses are extremely abundant across the world. They can actually infect all types of life on Earth, from going from plants to bacteria to vertebrates, even humans. But they can also infect protozoan parasites, and more specifically protozoan parasites that are able to infect humans and cause disease. And then you will get a kind of nesting doll model where you have a virus that infects a parasite, which in turn can infect a human. And within my PhD, within my project, I actually focus more on a fundamental question, fundamental evolutionary question, basically to understand the coevolutionary dynamics of these viruses that can infect and live inside protozoan parasites. And I use the Leishmania virus symbiosis for this. This is one of the best known virus-parasite symbiosis models. And to give a quick background about both of these organisms, I can say about Leishmania, there are protozoan parasites of the trypanosome family, and they cause the disease leishmaniasis, which can be considered as a spectral disease, developing various forms of clinical manifestations, going from visceral leishmaniasis, as you can see on the right-hand side, to the two middle pictures causing cutaneous leishmaniasis, which can be either locally or diffuse form, and even the more severe form, which is depicted on the far left, the mucocutaneous leishmaniasis, where the disease actually migrates to the mucous membranes and, causes a lot of scar tissue development over there. Leishmania parasites, they are transmitted by phlebotomin sandflies, tiny mosquito-like species, and they basically transmit through a more sylvatic transmission cycle, so going from the sandfly to a wild mammal, and then going back. And humans are basically considered as dead-end hosts within this major transmission cycle. But in more urbanized or peri-domestic settings, this transmission cycle can also change a little bit, where more synanthropic animals such as dogs, cattle, rats actually can also pose as a reservoir for these parasites, and even humans can become more reservoir species. Going over to its symbiont, Leishmania viruses. These are double-stranded RNA viruses with a small genome, 5.2 kilobase pairs in length. They only code for two genes, one capsid protein that encapsulates actually the double-stranded RNA, and then also one RNA-dependent RNA polymerase. And the virus is basically able to transmit to different parasite cells through vertical transmission, basically to cell division of the Leishmania parasite. But more recent research also highlights a horizontal way of transmitting, basically that the virus is able to hitchhike on the exosomal cell signaling pathway of the parasites, being able to infect uninfected cells through the exosomes. And so why it's important to not forget about this small virus, this small double-stranded RNA virus, is because the presence of Leishmania virus can have a big impact on the eventual pathology in the human host. It can cause more severe disease manifestations. So from the cutaneous lesions that I've shown, it can even migrate to the mucus and cause mucocutaneous leishmaniasis. So it becomes more severe. But also the presence of this virus has been associated with the treatment failure and symptomatic relapse of patients. So coming back, I focus more on the fundamental evolutionary questions to better understand how actually these viruses and parasites co-evolve together, or were able to. And basically, we have a general workflow for this to bring parasites into culture, into liquid culture medium. At ITM, we have a big cryobank where we're able to actually resurrect old samples of Leishmania parasites from different locations throughout South America. So we bring them back into liquid culture medium. We sequence them, we do a nucleic acid extraction and send them for whole genome or whole transcriptome sequencing. So basically what we can do is extract the DNA and try to extract the parasite genotypes. We can extract double-stranded RNA of the virus and do the same actually for the virus, or we can do everything together and extract the total RNA, where we can then use the reads and with a reference-based approach or try to extract the parasite genotype based on the whole transcriptome, and then with a de novo assembly approach actually try to assemble the viral genomes, and then sparing costs and time by just only doing one sequencing reaction actually catch both. So what did we find? We were looking for either looking at subgenus level of Leishmania, which is endemic for South America. And afterwards, we also dive into one specific Leishmania species. And for the subgenus, we basically found a topological cophylogenetic signal between the two most prevalent Leishmania species within South America. On the bottom phylogenetic tree represents the phylogeny of these two Leishmania parasites, with in gray Leishmania guianensis, in black Leishmania brasiliensis. And on the top, you can see the phylogenetic tree of the viruses, which actually already shows a nice concordance between the two, where you can see that LRV of Leishmania guianensis of the gray tips actually really nicely diverges separately from the ones of brasiliensis, namely on the black tips. But this is only for two species, while there are multiple species endemic for South America. So the next goal is to scale up the number of species, scale up the number of individuals, to see and better to formally test as well with global fit and event-based methods to really confirm whether we have a cophylogenetic signal or we really have mirroring phylogenies and actually have phylogenetic congruence. That's a project that's still ongoing. I'm now ready to assemble actually the viral genome, so very excited. But next to this subgenus research, I also focused on one species in particular, Leishmania brasiliensis, which is the main etiological agent of cutaneous leishmaniasis, the most important one, the most widespread, and the more generalistic species of this subgenus. What we find here, you can see a phylogenetic tree of Leishmania brasiliensis. All the tips here are positive for the Leishmania virus. But you can see in green, in purple, and in orange, basically three distinct ancestral populations that we depicted. And in pink and black actually represent individuals which show indications of intraspecific hybridizations sharing ancestry between these three distinct populations. And when we compare the Leishmania viral phylogenetic tree, we found 9 divergent viral lineages. And when associating this to the population structure of the parasite, we basically could find some interesting aspects. Namely, this can be summarized in the bar plot, where on the left-hand side you see the three first bars which represent these distinct populations. What struck us most was that the viral presence in these more isolated, distinct populations was much lower, with a viral prevalence of about 14 to 20%, maybe 26%, but that these intraspecific hybrids, which are caused by more secondary context of infected persons and synanthropic animals, actually gave rise to a much higher viral diversity, as you can see, and also a much higher viral prevalence, going up to 80% even. So yeah, this is something that's quite interesting to take into account as human migration increases at this moment in many communities, and that's so, as it so happens, parasite hybridization will probably increase, and such also the viral diversity will be maintained, and taking it into account these epidemiological consequences of the virus. This is actually a quite important result, and it was important to really look at these viruses and take these ones into account. So to quickly recap, on the subgenus level, we see a cophylogenetic signal between different Leishmania species and their respective viruses. We now want to actually see to what extent we have phylogenetic congruence, and we want to do this on the genus level, incorporating South American and more Old World Leishmania species and their respective viruses, and also on the subgenus level, with including more species. And then within specific species, we actually see also a cophylogenetic signal, but also a great difference between more distinct ancestral populations, which are more isolated, and then more recent hybrid populations, which has important epidemiological consequences. So with that, I want to thank the two teams who I'm affiliated to, more virus-related team, parasite team, all the collaborators that sampled back in the 70s, 80s, all these parasite samples, and FWO for making me stand here or paying my plane tickets. So yeah, thank you very much. I don't know if there are questions or time for questions. Yeah.
Maybe I missed it. How does the virus affect the Leishmania cells? Can they inhibit their growth?
Well, up until now, it doesn't really have been shown that there is an immediate thing going on with the Leishmania virus and the parasite, in the sense that it can be disadvantageous to have the virus. But it has found a way to stay alive inside the cell, and actually it mainly triggers hyper-inflammatory responses in the human host, eventually. Right. There are no other questions. I will leave it up for the last speaker. Thanks.
Good evening. I'm the last speaker of the day, so thank you all for sticking around. Looking forward to the poster session coming up soon. But my name is Gabriella Panayotakis. I'm just finishing up my first year in the Bhattacharya lab at Rutgers, New Brunswick. And today I'm excited to talk to you about work that our lab has been doing in Yellowstone National Park and how I will be continuing it for my PhD thesis. So we've been developing an in situ, in vitro, and ex situ geothermal bioreactor to model the evolution of multi-species interactions. Okay. So why is it important to do this work? So if you can understand how life was like back on early Earth, you can propose some, and habitability in general, you can propose some general rules for how stable ecosystems operate and how they're in themselves can be resilient. Yellowstone National Park is widely studied. I'm sure a lot of people in this room have done a PCR experiment, and Thermus aquaticus came out of Yellowstone National Park as a polyextremophile. The study system we're interested in at the park are the hot springs, and they're the most common hydrothermal feature in the park, and they're due to hotspot volcanism under the North American continent. So these hot springs have really diverse and complex microbiomes They span multiple kingdoms, holding prokaryotes, eukaryotes, and viruses alike. And the communities that survive and tolerate these systems often endure extreme temperatures, so very hot, extreme acidity, so low pH from like 0 to 4, solar radiation, so intense UV, and cycling of metals and other toxic elements in the environment. So why is this system interesting and why is it important to study it? It's generally thought that perhaps even though our Earth is mesophilic now, it started out as cradles of extremophilic pockets around the world and then later on became to what we know as mesophilic. So it's important to have a study system. Sorry about that, we started a little early. It's important to have a study system that has historically endured early Earth conditions and still endures similar inhospitable conditions. As I mentioned, there are many domains that live in the hot springs. The species that we're interested in are these extremophilic red algae called Cyanidiophysce. So there are 3 genera of these algae, Galderia, Cyanidium, and Cyanidia skyzon. They are about 2.5 billion years old, so they're pretty ancient, and they split from their mesophilic red algal ancestors 1,600 million years ago. The Galderia and Cyanidia skyzon are extremophilic, but Cyanidium tends to be more mesophilic. And in general, they follow the 1% rule for their genome, which says that they acquire 1% of their genes from horizontal gene transfer from environmental prokaryotes, which then confers their polyextremophilic traits, so their ability to live under extreme temperature, pH, and heavy metal cycling. A really important aspect to these algae are that they're the only known eukaryotic phototrophs that can survive in these inhospitable conditions. So if we can understand their persistence and how they're able to maintain a stable ecosystem in this environment, we can start to build universal rules for ecosystem resilience elsewhere in the future and potentially in the universe. So I mentioned these 2 algae, Galderia and Cyanidia skyzon, and although they co-inhabit the hot springs and take up about 90% of the biodiversity in the system, the physiology of the 2 still differ. So as I mentioned, they are eukaryotic, and Galderia, as you can see on the right, is a little bit bigger than Cyanidia skyzon, but also its metabolic preference is mixotrophic. So it can do either phototrophy or heterotrophy, whereas Cyanidia skyzon is primarily photoautotrophic. Sorry. In terms of their reproductive capabilities and dispersal mechanisms, they're also different. Galderia utilizes autospores and has a rigid cell wall, whereas Cyanidia skyzon does binary fission and has a very weak cell wall. And what we're also particularly interested in, so I mentioned that they have to tolerate intense toxic heavy metals, one of them being arsenic, one of the most pervasive and ancient toxins on Earth. And when you're dealing with arsenic as a microbe, you have there's a repertoire of arse genes that could be used to biotransform it and to allow you to survive. These algae differ in how they biotransform arsenic and how they persist in the environment. So Galderia actually is able to detoxify it to the point where the arsenic is no longer a threat. However, Cyanidia skyzon primarily effluxes it, so it takes in the arsenic species and then dumps it out almost immediately. So the divergent capabilities of these 2 algae, but their persistent coevolution in the system, and they're, as I mentioned, 90% dominant over other eukaryotes, prokaryotes, or viruses, leads us to led us to build these 3 questions. The first 2, which we have already answered, and the third we're continuing to work on. So the first question is, how do multi-kingdom interactions shape extreme environments like this one? Boop. Okay. So we did, back in 2021, we started to collect field samples from the Yellowstone National Park hot springs where these algae lives. We did genomic, transcriptomic, and metabolomic analysis on our samples. And what's important here to note, this is a picture of one of the characteristic hot springs, and you can see it's green, thriving with these red algae. And the locations we sampled from were rock, so specifically targeting the endolithic algae, soil samples, so soil that's nearby the stream that still maintains the populations of the algae, and biofilm, so sampled directly from the stream. Tim Stevens, a postdoc in our a postdoc in our lab, he did genomics work on our samples where he aligned the MAGs, the metagenome assembled genomes, to reference genomes for prokaryotes, eukaryotes, or viruses that could be in the environment, and specifically Cyanidea schizon morale and Galderia yellowstonensis. And what he found was that even though Cyanidea schizon seems to be the weaker microbe, right, less metabolically flexible, smaller, no cell wall, it was actually dominant in the environment based on the MAGs that we found. You can see this in the figure where Cyanidea schizon is represented by the red, and Galderia is pretty weakly represented by orange. And finally, we also noticed that prokaryotes are still fundamental to the ecosystem in the coloring here shown in the blue and the green. So this challenges previous assumptions that may have been brought up about the constitution of this ecosystem. It was previously thought that since Galderia is a little bit more robust, has a bigger genome, more metabolic capabilities, that perhaps it was the dominant alga, but it is actually the reverse, with C. morale being only photoautotrophic but maintaining its reign in the system. So then he analyzed the transcriptomic data, and the results confirmed the previous hypothesis that I just stated of what we think is going on with these two species. So again, the colors are maintained from previous, so Cyanidea schizon morale is seen in the red, and Galderia in the orange, and Bacteriarchaea in green and blue. And what we found that, again, is Cyanidea schizon is leading the RNA-seq data, with bacteria still having a strong foothold. But the important aspect here is that it's starting to answer some of the questions about why C. morale is dominant over Galderia. We found that diurnal cycling is a huge driver in the system. So during the day, perhaps Cyanidea schizon is dominant because it's photoautotrophic and it's the primary producer in the system. Whereas going into night, Galderia starts to peak and take some of the spotlight from the system with its heterotrophic capabilities, and this would explain why it continued to have these genes persistently, even though it's not dominant. The next question we sought to investigate was, how does gene sharing within this microbiome impact ecosystem resilience? So I mentioned HGT before, and as I'll reiterate again, the two alga in the system, Galderia and Cyanidea schizon, they got 1% of their genes from co-inhabitants in the microbiome, which is part of what allows them to maintain persist in this extremophilic environment. And a postdoc in our lab, Julia Van Etten, did work in monoculture with Galderia and investigated their arsenic resistance capabilities, and she found that generally, because arsenic, when comparing arsenic and mercury, which are both present in the hot spring, arsenic is a multi-gene response, right? The microbes have a few options of what they can do when they encounter arsenic based on the gene repertoire that they have. So perhaps they acquire the HGT from the environment, but because there may be a few genes going into their resistance mechanism, they need to keep the HGT and they can't just reacquire it easily later. So they highly integrate it into their metabolic profile, and this allows for reduced selective pressure on individual microbes because now the ARS resistance genes may be split up across the community. This is different from what happens when they encounter mercury, where it's only a single gene response to mitigate the stress. So perhaps the microbe can acquire the HGT, handle the mercury when it's present, and then if the stressor goes away, it also loses the HGT because it can just acquire it again at another point in time if necessary. So this model spells out the integrated HGT model, or IHM, in which in terms of arsenic, you may have a more complex stressor which allows for a community response in the ecosystem, and the benefits of this are that you can undergo gene neofunctionalization during those periods where the stressor isn't present, and you can split up the ARS genes among the microbiome community, allowing for individual microbes to have better resistance and health in the ecosystem, whereas mercury detoxification, it's very individualistic among the microbiome. This hypothesis was supported with the field sampling data that we did, where you see Cyanidea morale primarily expresses Ars-M and Galderia expresses Ars-H. And this is also in line with the diurnal cycling of when each of these are active. And we also found that Ars-M and Ars-H in general in these, the field sampling that we found, they have displayed loss of function, which indicates potential neofunctionalization going in line with the IHM model. So the next question that we developed for the system was what abiotic or biotic factors may underpin this complex polyextremophilic microbiome. We intend to do this by using an ex situ geothermal bioreactor, so essentially trying to combine the benefits of a lab monoculture, which we used to develop the IHM, and the benefits of field sampling that went into the first aspect of the questions. And this is important because when you have a monoculture, it's not exactly, it doesn't represent the reality of what it's like to live in the field. And when you're doing field sampling, it's difficult to change environmental conditions. So you don't get to see what the direct effect is of individual variables. So with this bioreactor, the main components that we thought were important to start with is have the algae in these flasks or these tubes, and have the soil and media and just Galderia to start, to try and equilibrate it to the Yellowstone National Park hot springs. Some questions that we intend to look at are how our microhabitat variation, how does that affect how does that affect the coevolution of these two algae, and what's different between the microhabitats. And some factors that we're planning, some variables we're planning to change, like I mentioned, are microbial community composition, arsenic stress, light intensity. These are all things that would be difficult to change with field samples, but are probably, you can attempt to do them with a bioreactor of this nature. So we started acclimating them to the soil already in the tubes, and you can see on the right that the Galderia in the bioreactor are more sparse than Galderia in a simple monoculture. And this is important to note because if you're doing lab experiments with the monoculture of these algae, you may think, because of their density and robustness, perhaps they may be responding differently than how they would actually respond in the field, where you see among these like soil particles, it's a little bit more dispersed and less populous. So finally, what we plan to do with the bioreactor once we equilibrate it to Yellowstone conditions, first we intend to do transcriptomics to track changes in the Ars gene expression of both species under arsenic stress or ambient conditions, metabolomics to measure the arsenite-arsenate abundance to try and understand how the biotransformation is happening and what's affecting that, and to investigate what is driving the metabolic preference of Galderia in this system, because it is a mixotroph and it has the opportunity to do phototrophy or heterotrophy, but Cyanidea schizon pretty much takes over. So thank you all for listening. These are the collaborators that have helped us with this work over the past few years, and I will take some questions. Yes?
When the HGTs took place, are there specific hotspots in the genomes where they integrate, and also do they acquire promoters for their gene expression patterns?
His question was about where the HGTs are located in the genome, and I'm not too familiar with this, but as I said before, they generally follow the 1% rule, so they're going to acquire genes that are helpful for them to sustain in these conditions. So genes like Ars-M and Ars-H would be considered HGTs for the alga. Okay, thank you.